Good morning and welcome to the Buck Institute for Research on Aging. My name is Mary McEachran. I'm the Chief Administrative Officer and General Counsel of the Institute. We're very happy you're here this morning and hope you'll come back to many more public seminars. The seminar this morning is co-sponsored by the Buck Institute and by CIRM, the California Institute of Regenerative Medicine. Many of you remember the initiative, the voter initiative that created the California Institute of Regenerative Medicine, and it has put California on the map in terms of stem cell research. And we are very grateful to CIRM for providing a matching grant for one of our buildings in which a great deal of stem cell research takes place, and a great deal of what you'll be hearing about this morning takes place in our CIRM-funded building. So many thanks to you as voters, and many thanks to the California Institute of Regenerative Medicine. I want to get in one plug when I said I hope you'll be coming back for more seminars. We have an open house coming up on November 26th. That's the Saturday after Thanksgiving. It's going to be fabulous. We'll have 3 faculty seminars on stem cell interventions is one, on breast cancer, and on Alzheimer's interventions. In addition, we're going to have great fun hands-on science for the kids. So while you're listening to the seminar, the kids or grandkids can be doing hands-on science. My role this morning is simply to introduce the speakers, the first 2 speakers. Kevin McCormick is the Communications Director at the California Institute of Regenerative Medicine. Before taking that role, he was in TV journalism for 20 years, many of those years here in San Francisco, and he considers himself the official translator for CIRM. When I called to ask him what else should I say, thinking he would give me his educational credentials, he said, "Oh, just tell people I'm terrified of public speaking," which I'm sure is not true. The second speaker of the first panelist is Karen Ring, and I'm very proud to say she is one of our alumni. Karen started the blog here at the Institute. It's called SAGE, Science of Aging, and I hope all of you will seek it out because it's a great source of information on new discoveries in age research. Karen went on to CIRM to head up their blogging there, so we're very proud of them. Kevin and Karen.
Okay. Good morning, everyone. How are you? I'm Kevin.
I'm Karen.
And first of all, I'd like to say how honored and delighted we are to be here today. It's always great coming to the Buck. In fact, as my wife was saying when we got out of the car coming in from San Francisco, she said, "Oh wow, they have fresh air here." And also I want to apologize for stealing Karen, but she did such a great job on the Sage blog that we wanted to have her come and do the same thing for us at CIRM. And is this, let's see, fancy. Oh, yes. Well, okay, let's begin. Our mission at CIRM is very simple. It's to accelerate stem cell therapies to patients with unmet medical needs. Everything we do is about the patients, and we always start every single talk that we do. Everyone at the agency uses this slide because it's a reminder of us why we're here. I think so often it's easy to get caught up in the day-to-day activities of your job and your work, and this reminds us that this is why we're here, the patients. And unmet medical needs is a particularly important part of what we do. This little girl is a prime example of an unmet medical need. This is Evangelina Padilla-Vaccaro, and she's with her mother, Alicia. They're from Corona in Southern California. Now, Evangelina was born with a really rare condition called SCID. It's severe combined immunodeficiency. It's also known as bubble baby disease because children with this condition have no functioning immune system. Even a simple cold could kill them. So in the past, they often had to live in a sterile plastic bubble to survive. This photo is particularly important because this is the day that Evangelina was cured. There's a doctor at UCLA, Don Cohen, and he had developed a method for taking blood stem cells from Evangelina, genetically re-engineering them to remove the SCID, the gene that caused SCID, and then reintroducing those blood cells into Evangelina. It created, as stem cells do, a new blood system and a whole new immune system, and she was cured. This is a picture taken about 3 months after the procedure. It's Evangelina and her mum and her twin sister and dad. Evangelina is one of 23 children that Don Cohen and his team have cured of SCID in the last 5 years. For a rare disease, that's an extraordinary thing. And now Don Cohen and his team are going to be using that same method, that same approach, to treating sickle cell disease, which is a disease that affects around 100,000 people in this country, and predominantly African Americans. And he started that clinical trial, and CERM is funding it. This is something I like to do, which is like, what are stem cells? Well, stem cells, they're like snowflakes that make up the snowman. Every single part of the snowman is made up of snowflakes, and every single part of us, from our heads down to our toes, all our organs, every single part of us is made up of stem cells. And to talk a bit more about that, here's Karen.
Thank you. This is my favorite subject. So, in my PhD and my postdoc, I studied stem cells. So I'll give you a little bit of background before we talk more about the applications that scientists are using stem cells for human health and disease. So what are stem cells? These are really important cells. They're unique in that a single stem cell can both replicate itself and it can differentiate into other cell types in the body. So if you were thinking about a skin cell can only become a skin cell, but a stem cell has the ability to become different types of cells in different tissues and organs. And so there's two main types of stem cells. There's an embryonic stem cell and also an adult stem cell. So let's discuss the two. Embryonic stem cells are also known as pluripotent stem cells. They have the ability, pluripotency means that this cell has the ability to become almost any cell type in the body. So any cell in maybe your liver, your heart, your brain. And how you can derive embryonic stem cells is that they start from, you start with a fertilized egg, which then develops into an 8-cell embryo. Let's see if this laser works, sorry. And then that embryo then develops into a blastocyst. At this stage, the blastocyst has something called an inner cell mass. It's this dark little area with a ball full of cells, and you can extract cells from the inner cell mass and culture them in a petri dish. These pluripotent stem cells can then become, as I mentioned before, different cell types within tissues like neural cells or neurons, cardiac muscle in the heart, or blood cells for your immune system. Adult somatic stem cells are different in that they're more defined. So they're different locations of these stem cell populations within different tissues and organs in your body. So your brain has its own set of stem cells, skeletal muscle, your blood, your liver, and your bone marrow all have different types of adult stem cells. And these guys are unique in that they can only become cells within that type of tissue. So you wouldn't have a brain stem cell that would become would be able to generate liver cells, so that's not possible. And then one other stem cell type that scientists are using now is an engineered type of stem cell that's very similar to embryonic stem cells. So shown here on the left is Shinya Yamanaka. He is a very famous scientist from Japan, but he also has a lab at the Gladstone Institutes at UCSF. And he is a really intelligent person and discovered a technique called induced pluripotent stem cells, which he actually got the Nobel Prize for in medicine in 2012. And these cells are very similar to embryonic stem cells. They can become any cell type in the body, but how you make them is very different. So to make an induced pluripotent stem cell, you can actually take skin samples from patients, grow the skin cells in a dish, and then you can genetically manipulate them by expressing these 4 transcription factors or genes that basically make up the identity of a pluripotent stem cell. So if you introduce these 4 factors into a skin cell, it reprograms that cell back to a stem cell-like state. So it says, hey, you're no longer a skin cell. We actually want you to go back to a pluripotent stem cell state. And from there, then you can make all the other cell types that you're interested in from these induced pluripotent stem cells. And this is a very powerful technology, as you can imagine, because you could make iPS cell lines from patients with specific diseases like Parkinson's or Alzheimer's, and then make these stem cells and then make brain cells from that and kind of ask, well, what's going wrong with these patient cells in a dish? And a question that I get a lot from the general public and patients is, well, how different are these induced pluripotent stem cells to embryonic stem cells? They're supposed to be the same, but are there any differences? And the answer is scientists are working on this question, and there are some differences still, and they're trying to work as hard as they can to develop technologies where iPS cells are exactly the same as embryonic stem cells. They just want to make sure that the stem cells that they're making are safe, that they make good stem cell models in addition. If they were ever to develop cell therapies from iPS cells, they want to make sure that when you transplant these stem cells into patients that they don't cause any tumors. So we always like to show this slide too. What is the potential and the power of stem cells? So, this parent is telling their child stem cell that they can be anything that they want to be when they grow up. And so one thing I like to talk about the power of stem cells is that you can model disease in a dish. And then we'll go into some stem cell therapy clinical trials. So one example that I like to give is this diagram showing how you can take skin cells from a person that has epilepsy, which causes seizures. It happens when there's the neurons in your brain fire too many electrical signals and they become erratic. And so you can take skin cells from a healthy individual and then skin cells from somebody with epilepsy and make stem cells using this iPS cell technology I just told you about. Develop brain cells from both of these people, and then look at their electrical activity. So on the left here, on the control, you see that these neurons fire once in a while, periodically, but that the epileptic patient neurons actually fire really, really frequently. So you can see the same types of cells side by side. The epileptic patient's cells are firing too many times. So what you could do as a scientist is say, okay, are there drugs that I can treat these neurons with to try to correct this epileptic activity and turn it back into the control? So that's one way scientists are modeling disease in a dish. And also, I also like to share this story because I think it's so cool, but these are cardiac myocytes, or heart muscle cells, that were derived from embryonic stem cells in a dish. So you can take embryonic stem cells and culture them in specific media, and a couple weeks later, you can generate these beating cardiomyocytes. Yes, movie works. So you can see them with your naked eye. They're just beating in synchrony together. This is really powerful because not only could these cells be transplanted into mice that have heart disease to try to help cure some of the damage that's happened to the heart, but as again, you can, as I mentioned, you could test different drugs to see if they help symptoms of heart disease, or even test toxicity, see if certain drugs might be toxic to these heart cells. So now I'll give it back to Kevin.
Is this working again? Can you hear me all? Okay, great.
I can hear you now.
So as Karen was talking about, we have a number of different cell types that we work with, and one of the ones that we started with was embryonic stem cells. Proposition 71, the voter-approved initiative that created us, was basically a response to the federal government's ban on the use of federal funds for stem cell research, embryonic stem cell research. And so the voters of California basically gave a giant kind of thumb in the nose at President Bush and created CERM so that we could do this kind of work, that we could create facilities like this here at the Buck to do some really important work. When we talk about embryonic stem cells, the embryos that we use or that are used in this kind of research actually come from IVF clinics. And so what they do is when couples are going for having trouble starting a family, they'll go to an IVF clinic to get some help. The woman will donate dozens of eggs, they'll be fertilized, and then they'll use them to have children. But when they reach a point where they've stopped, they have enough, and they don't need to use any more eggs, they're faced with a choice as what to do with those embryos. And as Karen said, these are tiny, these are about 5 days old, so they're very small. And so they have a choice. They can either donate them to another couple, and that happens in maybe 2% of cases, or they can destroy them or donate them to research. And so that's where the embryos and the embryonic stem cell lines that we talk about, that we use in research, come from. And we use them in a number of different ways. We ought to use them to be able to create brain cells that may be able to help people who have Alzheimer's or Parkinson's, bone cells to help repair damaged injuries or osteoporosis, and also maybe the pancreas to help people who have type 1 diabetes or type 2 diabetes. And so this is what we've been doing with a company called ViaCyte. This device here, this is what they developed, and it's a pouch, and as you can see from that's a quarter. So you can see how small this is. This is actually about as long, as wide, and as thick as a credit card, but only half as wide. And it's implanted under the skin at the back. It's a really simple outpatient procedure. And it's filled with embryonic stem cells that have been turned into the kind of cells you find in the pancreas. And so what this device does is it's under the skin in the back, and as blood flows across it, it's able to measure blood sugar levels. And when those levels are abnormal, it's able to secrete insulin to be able to correct them. And the hope is, and this is in phase 1 clinical trials now, we're just testing this. The hope is that if you can develop something like this that can be implanted under the skin, you'll completely change the life of people with type 1 diabetes. So instead of having to constantly measure everything they eat, constantly check their blood to see how they're doing, inject insulin several times a day, this would take away the need to do that, effectively curing them. And it would also work for many people with type 2 diabetes. And this is tremendously important because diabetes is not just a huge problem here, it's a global health epidemic. And if we can find something that helps so many people who have it, that would be extraordinary. Now, another way we're using embryonic stem cells is in a spinal cord injury trial we're working with a company called Asterias. This young man is Jake Javier, and he's from San Ramon on the other side of the Bay Bridge. Earlier this summer, Jake had graduated high school, was getting ready to go to Cal Poly on a scholarship. And so he went round to a friend's house for a party, a pool party. He jumped in the swimming pool and broke his neck. And he was paralyzed from the chest down, had almost no movement of arms or hands. And he enrolled in this trial, and they transplanted 10 million stem cells, embryonic stem cells that have been turned into a cell called oligodendrocyte. They transplanted those into his neck, and the hope is that those cells will migrate to the injury site and create a communication, essentially restoring communication, a bridge from Jake's brain back to his body. Within a few weeks, I saw Jake about 3 weeks after the procedure, he was able to move his arms, and he had some feeling in his hands, and he's making more recovery since then. So that's pretty impressive, and even those small gains are important because it gives someone a real sense of independence that they wouldn't have had before. If you think Jake's progress is impressive, then this is Chris Boson, who had exactly the same problem as Jake. Chest, paralyzed from the chest down, almost no movement in his arms and hands, and he was the first patient actually to get the transplant of 10 million cells, and he's able to move his arms, as you can see, use his hands, and this has given him a whole new sense of possibility of what his life could be. Now, something like this, it's important to say, can happen spontaneously in people who've had a spinal cord injury. About 1 person in 5 will experience this kind of recovery. But all 5 patients who got 10 million cells transplanted had progress. Some, like Chris, went up what we call 2 motor levels. Most of the others went up 1. But even so, the fact that all 5 saw such improvements is really encouraging. And particularly because this is only the 10 million cells is considered a half dose. They're now starting to work with patients with 20 million cells, and that's what they think will be the therapeutic dose. So the first couple of cells, the first couple of patients they work with, it's all about testing for safety, making sure that this isn't going to harm the patient. Now they're looking to see how much benefit this may have. So we're getting into some pretty exciting stuff. Then we talked about adult stem cells, and these aren't as flexible in some ways as embryonic stem cells, but they're still amazingly powerful. This woman is Sonia Alia. A few years ago, she had a stroke, and it left her with really impaired speech and very limited movement on her right side, her right arm and her right leg. And she's still a young woman, and so she said at the time she really just thought she wanted to die, because life as she knew it was over. Then she enrolled in a clinical trial at Stanford with Dr. Gary Steinberg, and they took donor bone marrow cells that had been modified to help boost their ability to help cells in the brain, and transplanted them into Sonia. Within a few weeks, she was regaining her speech and was able to use her arm and her leg again. Now, this is really incredible because for 2 reasons. Number one, this was a phase 1 clinical trial. And in a phase 1 clinical trial, you're basically just looking to see that this is safe because you're doing something that may never have been tried in people before, using cells in the brain that had never been tried before. So the researchers are just seeing, is this safe? That it doesn't do any harm, it doesn't make the person's condition worse. So the fact that, and they often use small numbers of cells to do that. So the fact that we're seeing this kind of improvement is really impressive. The second reason is that many of the patients, and there were 18 altogether in this clinical trial, all of them experienced some benefit. Some, a few as dramatic as Sonia's, some not so dramatic, but still they were able to do things that they hadn't been able to do before. Some of these patients had had their stroke 3 years earlier. And in the past, it was always felt that if by about 3 to 6 months after your stroke, the level of recovery you had made was the level of recovery you'd make. You wouldn't go any further. And this showed that you could actually rewind the clock in a sense. You could regenerate the brain to get it to be able to do things that hadn't been done before. That's an incredible finding. And now the researchers have started a larger clinical trial, and they're going to be using larger numbers of cells, and so that's already going on. Another researcher we're working with is Henry Klassen and his team at UC Irvine, and they're working on a disease called retinitis pigmentosa. It's a relatively rare form of blindness. So the image at the top is what we would normally see. With retinitis pigmentosa, what it does is it starts to destroy some of the cells at the back of the eye, the retina. And so it kind of creates this tunnel vision. It squeezes and squeezes until there's nothing left. It's a really devastating disease, and it often strikes people quite early on. So what Henry and his team have done is they've taken adult stem cells and then taken them into the lab and turned them into what are called retinal progenitor cells. And these are the kinds of cells that are found in the back of the eye and the retina, the ones that are being destroyed by this disease. So they turned them into the retinal progenitor cells, multiplied those in the lab, and then re-injected them back into the patient. And they've just completed the first data safety monitoring thing, and the first 9 patients all did well. No complications, no side effects, which is very important. And this woman, Kristen McDonald, we got to see her a few weeks ago. She was one of these first patients, and she said she's able to see shapes, things she wasn't able to see before. She can't really see very clearly, but she's seeing things that she wasn't able to see before. And so it's really encouraging. And again, this is a phase 1 clinical trial where they're just looking to see that it's safe. And so to actually begin to see some kind of benefit at this early stage is really encouraging. And we're funding a similar clinical trial with a researcher at USC who's working on macular degeneration. And that's really important because that's the leading cause of vision loss in the US. And then what iPS cells, as Karen was explaining to them, I think these are the coolest ones of all because in a sense they're man-made. These are things that we use ourselves. And she talked about, you can take these cells and turn them into any other cell in the body and they have the same characteristics as that person's disease. And one of the researchers we work with is Alison Motri at UC San Diego, and he's doing the Tooth Fairy project. What he does is he gets parents to send them the children's teeth. And so he collects teeth from children who have autism and children who are healthy. And then he takes some of the cells from those teeth, turns them into brain cells, and he's able to do a cross comparison, seeing what the brain cell of a child with autism looks like compared to the brain cell of a child that's normal and healthy. And he's able to identify differences and figure out, okay, what's going on here? And then, as Karen talked about that whole notion of drug screening, he's able to think, okay, what compounds, what drugs, what medications that have already been approved by the FDA are out there that we can then use to help some of these people. So it's an extraordinary tool. And I think increasingly with IPS, even though it's a relatively new discovery, this is going to be something that more and more people are focusing on.
So staying with the iPS cells, currently that's the CERM is funding more preclinical work using iPS cell technology since it's such a new technology, but there are some scientists who are testing cells derived from iPS cells in clinical trials, and one of them is for macular degeneration, as Kevin mentioned, a disease where you lose sight because the macula in the eye starts to degenerate. And so this woman, Masayo Takahashi, who's actually worked with Shinya Yamanaka before, was able to develop progenitor cells from the eye, and she makes these sheets, and you can see that they're pigmented. They're called pigmented epithelial cells, and she can develop this sheet of cells from stem cells and then transplant them into the patient's eye, and these cells hopefully will integrate and help the patient start to see some sight again. So this trial was started about 2 years ago, and it was stopped because they were concerned about the iPS cells having a genetic mutation that was caused by the iPS cell development process. But since then, they fixed that issue and they've restarted the clinical trial. So it'll be really exciting to see, whether this technology will help patients. We hope it will. And CERM is looking to fund future projects such as this. Oh, okay. So CERM is actually funding so far has funded 21 clinical trials, and our goal in the next 4 years with our $800 million that we have left is to fund a total of 50 clinical trials. 10 of these clinical trials were from projects that we had supported in the early stage, and then they developed into clinical trials. And all the trials that we're funding cover a wide variety of diseases, including cancer, heart disease, brain disease, diabetes, etc.
So as Karen said, we're funding a lot of clinical trials. In the first 10 years of our life, we funded 10 clinical trials. We now plan on doing another 50 in the next 5 years. It's pretty ambitious, and we're working very aggressively to try and recruit all the best science and the best scientists that we can to do this work, because it's very important and a key part of our mission. But one of the biggest problems we face in doing this is the FDA, the Food and Drug Administration. The FDA has regulatory approval over everything that we do. And now the FDA does a really good job on many levels. It's a very difficult job to do, because if they approve something too fast and there are problems, everyone jumps on them. If they approve things really slowly, the people who need those therapies jump on them. So they're kind of stuck in the middle. And part of the problem is that they've kind of developed this policy of doing as little as possible. In the last 5 years, since they introduced 15 years since they introduced new regulations that were supposed to simplify the approval process, they certainly simplified it because they simply haven't approved any stem cell therapies. Now, when you think about the need that's out there, I mean, clearly it may be that in the early days the science was still relatively new, and so there weren't many great projects that were worth doing. Today, that's certainly not the case. I mean, as you saw some of the stuff that we're working with, there are great scientists out there doing great work, but the FDA has a process of slowing things down. In fact, they've created probably the most kind of burdensome regulatory approval process for stem cell therapies that exist anywhere in the world. Far more complicated than for traditional medications. And there may be a reason for that, in the sense that the FDA evolved, regulating normal medications, ibuprofen, Lipitor, all the things that we're used to hearing. Regenerative medicine is very different. A typical medication goes in and passes out of the body. With stem cell therapy, we're hoping these cells will stay in there, sometimes for a short while, several months, sometimes indefinitely. And the idea is that they will help completely regenerate something. So the FDA is struggling on how to deal with that, how to regulate that in a way that still maintains patient safety, but advances the science. And right now, the balance it's not in the patient's favor. Remember this little girl, Evangelina, who was cured of SCID? Well, Don Cohen and his team at UCLA are using the exact same method that they used to cure Evangelina and 22 other children to treat people with sickle cell disease. And the FDA gave them approval to start a clinical trial. But they said, after you've treated the first patient, we'd like you to wait 6 months, just to make sure it's safe, that there's nothing going on. And so they're having to wait 6 months using the same technique that they used in children born without an immune system to see if it's safe. For the researchers, that's really frustrating. And for the patients who are waiting for these therapies, it's intensely frustrating. Patients with sickle cell disease can probably wait. 6 months is not too long for them, not too burdensome. But if you think about someone with ALS, Lou Gehrig's disease, the average life expectancy for someone with ALS from the time of diagnosis to the time they die is between 2 and 4 years. To have to wait 6 months for treatment could be a death sentence. So clearly something has to change. And one of the things we're trying to do at CERM is start this Stem Cell Champion campaign. We're trying to recruit as many people as possible, patient advocates, people like yourselves who are interested in science, everyone who has an interest in advancing this, in making things better, making things move faster, because we have a real need to move things through much faster than they do right now. Right now it takes a traditional medication about 3 years to move from being a good idea into a clinical trial. For stem cell therapy, it takes 8 years. For a traditional medication, it takes about 10 years to get through the whole clinical trial process and into the market. 10 years and up to $2 billion. For a stem cell therapy, we don't know, because the FDA hasn't done anything about that. And so for people like Katie Jackson, this is a problem. A few years ago, Katie was pregnant with her 3rd child, and they found out that her husband, Mike, had Huntington's disease. This is a nasty neurological disease, always fatal. There's no treatments and no cures. Mike, it's hereditary, but Mike didn't know he was at risk because he didn't really know his father. And it was only after Mike was diagnosed that he learned his father had Huntington's. So Katie went from having a husband and 3 healthy children to a husband who had a fatal condition and 3 children who had a 50-50 chance of developing this. So we need to, that's why we did Stem Cell Champions. We want to get as many people as possible to work with us to help move the FDA, to help get them to change the way they work, to create a more efficient, hopefully faster, but no less safe approval system. Because for people like Katie and her family, their lives depend on it.
So if you're interested in learning more about our campaign, we actually have a really fun short video on our website at cerm.ca.gov stem cell champions that tells you more about the campaign. You can also sign up for our email list and spread the word. If you want any more information, this is our contact. We have our website, our blog, which has a lot of great resources about the latest stem cell research and some of the studies we're funding. We're also on Facebook, Twitter, and we have some really cool YouTube videos. So thank you very much.
And as you're leaving, what I'll do is I'll have some of these sign-up sheets out there. So just your name and email address. And so if you're interested in joining us and learning more about how you can be a stem cell champion and how you can help us create the kind of change that we all need at the FDA, then these will be outside for you. So thank you very much.
My name is Christopher Wiley. I am a postdoc, let's see if this works, okay, in Judy Campisi's lab here at the Buck Institute for Research on Aging. And what you just heard about are some of the really exciting therapies that are being developed where we make cells good, where cells are getting better and we can actually regenerate people. I want to tell you a little bit more of a story about what happens when good cells go bad. So I'm going to talk to you a little bit about this in a story that surrounds HIV, which of course is a big plague onto society right now and a major health concern for many people. But I'm going to tell you a story that's a little bit different than probably what you're used to hearing. So for those of you that don't know, HIV is the virus that causes AIDS. Most people don't get AIDS anymore. HIV, though, is still a big problem. It's still a large problem in our society and around the world. It's especially growing in third world countries. And this here is a little image of an artist rendering of an HIV cell. The problem with HIV is that it infects the immune system. And how do we get rid of viruses and other diseases? Well, our immune system gets rid of them. So when HIV gets involved, it's a very dangerous problem because now you can't get rid of the biggest problem you have. So this is what a health span looks like of you, me, any normal person. If you're a normal person today, age 25, chances are you're better than 50% that you're gonna live to be 70 or older, okay? Back in the day when people were first getting HIV and there was no treatment, no cure, no anything, people would develop AIDS and they would die very quickly, so your lifespan looked a little something like this. So this is dramatically shorter than what you would normally expect. The good news is we now have treatments, we now have suppressive therapies, and they work like this, and you can see that we're moving people's lifespans longer. The green arrow is pointing to where we're going now. The thing is, people aren't getting AIDS, even though they're getting treated, and they're not going back. They're not living as long as the rest of the uninfected population. They're living in this intermediate zone. So something else is going on here that's beyond AIDS that's causing a problem for these people. And what we think might be going on is that these people are developing conditions that look like premature aging. They're actually looking like they're 5, 10, 20 years older than they actually are. And a lot of the conditions that they get associated with seem to be things that might be driven by the medications used to suppress HIV. Among them are loss of fat from the skin, excuse me. So this is a common thing you'll see with HIV patients is you can see here and here, they lose fat, subcutaneous fat here in their face, so they actually look like they have kind of a hollowed out expression. They lose bone mass, so it can become osteopenia or even osteoporosis. They have diabetes, often. They struggle to think in an Alzheimer's-like way, and they're also developing symptoms of heart disease. So in short, they're really looking like people much older than they should be for their age. And we really don't know why that's the case yet, but we have an idea. One of the things we found in the Campisi lab is that some of the medications that are used to treat HIV are actually causing cellular senescence. So cellular senescence is a process by which cells stop dividing and they become really more like an old cell. As we age, we get more and more of these. And people who are being treated with HIV medications might be getting even more of these than just you or me. And so here are just two examples of two older HIV medications that we've treated cells in the lab with. And blue is a marker, this blue staining you see is a marker we use for senescent cells. And we find that by treating just regular cells from your body with these HIV medications, we can actually find that they drive senescence, and therefore these cells are actually getting older. So one way I like to describe senescent cells is, if you think of a tissue like you think of blocks in a neighborhood or a city, and each cell is a house in that neighborhood, then a senescent cell is kind of like the old, rundown, dilapidated house. It's probably got a broken sewage system, there are rats living in it, it's falling apart. And now I want you to imagine, if you live next to that house, you're probably getting rats inside, you're probably getting sewer smell. This is not a place you want to live. And one house like that could ruin an entire neighborhood. That's what senescent cells do. They release all sorts of factors that can mess up the local environment, that can turn that tissue from a happy, healthy tissue into something that more or less resembles an aged tissue. Here's one example of what senescent cells can do. So we just took factors from normal cells or senescent cells, and we cultured fat stem cells in the presence of these factors. And normal fat stem cells, as you see here on the left, they will develop into fat cells, and we can stain them for red, so this red is indicating fat cells. And so you can really see that they'll become a normal fat cell. That's what they normally do. That's what fat stem cells like to do. They like to become fat. But when you culture them in factors from senescent cells, that doesn't happen. They stay in that state. Now, if you're thinking about this in terms of regenerative medicine, if you're old or sick, and you put your stem cells in or your derived cells, this is probably not what you want, the kind of milieu you want to put them in, because those secreting senescent cells can cause all sorts of problems. The good news is, we've now developed ways of eliminating senescent cells, and we're doing this in mice right now. So these mice are exactly the same age. This is a premature aging mouse that we've learned to test a way of eliminating senescent cells in. This is a mouse, aged mouse, that has not received any treatment, so it has all of its senescent cells. Here is a brother that has had its senescent cells removed. Look at that. See the hump on its back, the closed eye, there's some hair loss, all these things that we associate with age. This mouse here doesn't have it. And if I had a video, I'd be able to show you. It's running around, it's happy. The other mouse is barely moving and is shivering. It's amazing what the difference you can see between these mice. So what do we want to do next? Well, the first thing we want to do, there are a lot of different HIV treating medications. There are a lot of different ways of treating them. We want to see which ones drive senescence and which ones don't, because if there are ones that don't drive senescence, those are probably the ones that you'd want to have. If you had HIV, you would want to have treatments that would actually not drive age-related disease. And then the second thing is, if we get rid of the senescent cells, can we prevent HIV medications from driving that premature aging? And that's one of the directions we're hoping to go in the near future. And so I just want to thank a few people here. Judy Campisi is my advisor. She's the one that runs our lab. Sue Liu did the fat stem cell work for me. And Chisaka Kieneman is actually doing the NRTI treatments. She's a graduate student in the lab. I also want to thank all of you, because it's always fun to see all those people smiling at me when I get up here and give talks like this. So thank you.
Good morning. I'm a postdoctoral researcher here at the Buck Institute as well. I work with two different labs, a lab that works with fruit flies and a lab that actually develops stem cell therapies, or that studies the ways to improve those stem cell therapies. And what I've done is that I've been working on a project that combines these two things and actually helped us go on a way in which we made a discovery in the fly that we now can apply to the type of therapies that you just heard Kevin and Rain describe to you about. So we are an institute that studies aging, and one of the problems that happens with aging is that actually our tissues degenerate, they get damaged, and that's not good. And now you know that we have stem cells and that we can use those stem cells to repair those tissues. That's the promise that regenerative medicine has put on the table. So the problem is that stem cells cannot do the work alone. So the stem cells are put into these tissue environments that Chris just described to you. And these tissue environments are not always the best host. I'm sorry, I don't know. It's okay. So these tissue environments are not always the best place where these cells like to integrate. So there's different things that block the ability of these cells to go inside the tissues. And one important thing that I'm going to focus today about is the immune system. And these cells, which are the macrophages that exist in all the tissues in our body and that are circulating. So I have here some videos for you. This is what a macrophage looks like. You can see that it has these ramifications. It can touch different cells and actually modulate what is happening in the environment. And one of the things that these macrophages do is that they eat the cell debris that it's around, and that's a good thing that they can do. And when the tissues get damaged, which you can see by that circle appearing in there, these cells will actually get attracted to there. And they are going to be able to do two things. So either they are going to kill more dead tissue, or they are going to help regenerate that tissue. And this depends on what kind of macrophage they choose to be. So if they choose to be a pro-inflammatory macrophage, they are going to kill more tissue, and they are not going to help to regenerate it. If they decide to be an anti-inflammatory macrophage, they are actually going to behave like a doctor and actually help glue things together and make a tissue functional again. Okay, so when you have a tissue with a crippled house like what Chris showed you, what happens is that the bad guys like to hang around those neighborhoods, and they are going to be terrible to make a regenerative therapy work. Though those type of people can change and become doctors. And if we can do that, then they will help actually to heal those tissues. And what we are looking is into ways to make one into the other. So that's actually one of the places where we can intervene to increase the success of the regenerative therapies. Of course, you cannot do that alone, and when you do that, you also have a hope that you can delay the rate at which your tissues are going to damage, and that you can improve the stem cells themselves. So now, as Kevin and Karen told you, we have some stem cells in our body which can regenerate tissues. Those are the adult stem cells. But in many tissues, we don't. And that's the case of the retina, which they gave you some examples to. So the retina is in the back of our eye, and if the cells that sense the light die, they are not going to be formed again naturally. So luckily, we have a lot of very smart scientists that have found ways to use stem cells and transform them into these photoreceptors, which are the cells that sense the light, and we can put them back in, and you can see that there's already clinical trials that are happening for that. So let me explain you a little bit how this works. Think of your eye as an orange. And now think that you're injecting cells, which are those photoreceptors that we created in a dish, into that very thin layer that separates the peel from the pulp. That's where we put the cells, okay? If those cells find a healthy environment, those cells are actually going to move in. And those round green circles that you see within the blue are actually photoreceptors that we put in that tiny space and could move in and are integrating into your retina. So now, here, the scientists that work at the Buck, what we do is that we come up with the ideas that then clinical trials can implement to develop therapies. So we always start by thinking, what is the problem that we want to solve? And the problem that I wanted to solve when I got here is that we skipped one, is that I wanted to improve the success of stem cell-based therapies. And what was the problem that I thought that existed? Is that we know now that we can do these things, we can transplant those cells can integrate. But this is all going to happen in a good way if we do it in a healthy retina. But the people that we are going to treat, they are actually not, they do not have healthy retinas. They have diseased retinas that are degenerating and that are full of those bad guys that are going to be a roadblock for those cells to integrate. So we can find ways to transform the bad guys into the good guys, then we can maybe make more cells integrate and help the success of these therapies. So the first step in the science lab is always the discovery step. How are we going to look for things that can do this job? And when we want to do this, we turn into simpler organisms, so that we can run big screens and look for molecules that can do what we want. So we wanted a molecule that could transform the bad guy into a good guy. We had a fruit fly, which is a very easy system to work with where we can make very big screens. And we looked for molecules that when we would give to these fly macrophages, would transform them into the doctor-like ones. And what you can see that is that if we give this molecule, MANF, to those fly macrophages, they transform into the healthy ones, and they can make the damaged eye of a fly actually become more repaired, bigger. While if you take that molecule from those fly macrophages, then we will enhance damage and you have a smaller eye. Okay? So we had the first step of discovery. We moved into the second step, which is to see if this works in organisms that look a little bit more like us. I know you don't like to think as mice as organisms that look a little bit more like us, but they do. And so we go and we do this, we test these concepts in a mouse model. So we have mice which have retinal degenerations, and the first thing we did was we delivered this molecule to the eyes of the mice and asked whether that could stop the rate at which they were degenerating their retinas. And what we saw is that yes. So I'm showing you here again a slice of the orange, and what we are seeing is two things: that we have less dead cells, and that we have more cells sticking around and alive in this mouse in which its retina is degenerating. So on the left, the red staining, that the red dots that you can see is actually a stain that we can do in the lab that marks dead cells. And as you can see, when we give these mice these molecules that transform their macrophages into the good ones, we reduce the number of the red dots, which are these dead cells. If you look on the right, what you can see are actually the alive cells. And while on the mouse that is degenerating, you only have 2 or 3 rows, by giving this molecule, we could actually preserve like double the thickness of this row. So then, we moved into our initial hope, which is to ask whether now we could improve the success of these transplants that we do into the retina if we supplement these transplants with this molecule that changes the immune cells in the retina. So what we are seeing on the left is the transplant that I showed you in the beginning in this degenerating retina, where cells do not move in, they do not integrate. If now, in the exact same of mouse, we give the exact same of cells, but we add this molecule with it, now these cells are happier, they will move in, and they will integrate. And what's very important is that if we test now if these mice that in the beginning of the study can see anything, if we test if they can see, by testing if they can respond to light by generating an electric impulse, we can see that the blue line that you see there, which is the electric impulse generated by the photoreceptors of these mice, actually happens when the transplant is done together with this molecule. So what I want you to take from this is to see what the work that we do here at the bug, which is to start from a very simple discovery in a simple organism like the fruit fly, test it preclinically in a mouse model, so that we hope that sometime the people that work with Kevin and Karen at the CERM can translate into a therapy that can be used in you. And I just want to finish by thanking the people that did all this work with me, because the work that we do here at the Bug, we don't do it alone, we do it always in teams. And this is the work of 2 different labs, the Henry Jasper lab and the Deepak Lamba lab. And the people on the bottom are the people that actually did different parts of this work with me. So thank you for your attention.
Okay, good morning everyone. My name is Emilce Rodriguez. I'm a postdoctoral fellow in the lab of Henry Jasper. here at the Buck Institute. And today I'm going to be talking about how do intestinal stem cells deal with protein trash and why this is important for aging. So have you ever thought about why we keep a clean environment in our houses? So instead of having something like this, we try to keep it like this. So there are many reasons, but a very, very important one is that we want to promote a healthy environment. And similar to us humans, cells in our bodies have to also keep a clean environment. So in this picture, you have a cell that has a lot of protein trash in this case, and the cells invest a lot of energy into removing that trash, recycling it, and try to go back to normal and promote that healthy environment. But how can a healthy environment be achieved? So if we're thinking of that picture of a room full of trash and things that you want to recycle, you can think about trash in two different ways. So you can categorize it depending how complex this trash is. So for example, if you have a brown bag that you want to get rid of versus a more complex type of trash, which is like a big pile of trash. So depending on the size of this trash, you will use different sizes of bags. So for this small brown bag, you will use a small trash bag. To collect all of this pile, then you will use a bigger bag. Then eventually you hope to grab all of that trash and then recycle it to go back to your healthy environment. So very similar to, in our case, to humans, our cells in our body, they also have to deal with trash. But of course, trash that the cell deal with is not this trash that we deal with. It's actually protein trash. And I'm gonna explain a little bit of what our proteins are. So proteins, you can see them as Legos. They're known as building block of the cells. So this, you can imagine Legos building a house or something. So the cells actually make up the cell, the proteins itself make the cells. The good thing about proteins is that proteins are the workhorse of the cell, meaning that they move things around, they signal, they actually make the cell work properly. So you can think about it as a workhorse made by Legos. The bad things about proteins, is that they require a special shape. We call this a fold in order to work properly. So in this picture over here, you have a folded protein, and when this protein becomes unfolded, it cannot work properly anymore. So you can imagine as a Lego that's just been melted a little bit, and now it's not gonna work as you wanted it to work. The ugly part about proteins is that when they get misfolded, they can aggregate. So you imagine a bunch of melted Legos together, clumping together, and that's probably not good. You cannot do anything with it. So this is similar to what happened to protein trash in our cells. So you have folded protein means functional protein, unfolded protein means it doesn't work anymore, and aggregated proteins, a bunch of unfolded proteins clumped together. So similar to our case, then the cell itself deals with this type of protein very different in order to eliminate it. So if there's an unfolded protein, there's a machinery in the cell called the proteasome that takes this unfolded proteins and break it apart back into the building blocks and recycle it. And there's another type of machinery called autophagy, and which takes big aggregates of proteins and breaks them apart and recycle its building blocks. So the idea is that then you recycle back and then you go back to this healthy cell. But something that happened to us when we age is that our cells lose the capacity of cleaning up the mess, removing the trash. So you can imagine that when you have an accumulation of protein aggregates, that impairs cell function and then can also lead to cell death. And this has negative impact to our body because it can cause diseases and also can affect normal function of tissues, and I will explain why. So there's a group of cells, like Karen mentioned before, that are particularly sensitive to protein aggregates, and these are somatic stem cells. These are the stem cells found in the adult tissue. So you already seen this picture, but just to remind you, so we have stem cells right now in our body in many, many places. One of them is the brain, the skin, the muscle, blood, and the gut. So if you heard about that you lose a lot of dead skin cells, and the reason why you don't lose your skin is because in your skin you have a lot of stem cell continuously regenerating this tissue. And that's why after losing that many dead cells, you still have a skin. So there are stem cells dealing with that all the time, similar in the intestine. In the intestine, every time we eat, cells get lost and they're replenished by stem cells in the intestine. So stem cells are very important. But however, aging impacts stem cell ability to eliminate protein trash, but we still don't understand why, and we need to understand more. So how can we study the way autosomatic stem cells maintain a clean environment? We can do this many ways, but the way I do it is by study the stem cell in simpler organisms. So you already heard from Joanna about fruit flies. So I also use fruit fly in my research. I look at their stem cells. So basically, you may be wondering why we use fruit flies. So flies and humans, they share 44% of genes involved in tissue growth and structure. This is a schematic of the Drosophila intestine compared to the schematic of the mammalian intestine. And you can see that they do have a stomach, they do have an intestine, posterior midgut, and hindgut and everything. Also, the other things that are more technical and makes the fruit flies very useful in the lab is that they reproduce quickly, they're very easy to maintain in a lab, and there are many genetic tools available, and also discoveries made in flies can be translated to mammals, to us, which is similar to what Joanna was just saying. So in order to study how allosomatic stem cells maintain their clean environment, I'm using the intestinal stem cells found in Drosophila. So if you were to look at a fly, this red tube over here is the intestine. If you dissect the intestine, it will look like this, very simple. So basically we tend to look at this posterior midgut part, and then if you look at the surface of the intestine, this is a cartoon representing that, and it's very simple epithelium. It's only formed of 4 different cell types, and the green cells over here are the intestinal stem cell. The really cool about these cells is that when they divide, they give rise to all the other cell types in the tissue, so they're very important for maintaining the tissue healthy. And that's a cartoon. This is how it actually looks. This is a picture in the lab, and you can see that you can label using molecular tools. You can label the cells in different colors, so you can identify them really easy. And here in green are the stem cells. So the type of work I do is that I do some genetic tricks in this intestinal stem cell that I fill them with aggregates. In this case, I want to see the aggregates, so I label the aggregates with red fluorescent protein. So just by looking at them in the microscope, I can see the stem cell full with aggregates. So this is what you're seeing here. So this is a surface view of the intestine, and you can see these red circles are the stem cell filled with trash. In this case, it's red trash. So I fill them with this trash, and then I look at later time points to see whether this stem cell from young animals are able to eliminate them. And the answer is yes. So initially you see the stem cell full with this red trash, then eventually they eliminate them. And that's very exciting. However, if I do exactly the same experiment in all flies, all intestinal stem cells fail to eliminate the trash. So this is another view of the surface of the intestine. In white here are the aggregates, and you can see that even after 2 weeks later, I still find the aggregates, meaning that the stem cells cannot eliminate them anymore when they get old. But there's good news, and the good news is that you can test different drugs in these animals, and this is a drug actually made for humans, and I tested in the flies to see whether I could improve the elimination of these protein aggregates. So basically what you do is that you take a lot of old flies and you fill their intestinal stem cell with this trash, and then you feed them with or without the drug, and then you see whether the drugs actually improve the elimination of aggregates. And that's exactly what I see here. So this is an old fly with the intestinal stem cell carrying these white aggregates, And after I treat them with the drugs, I see a reduction in aggregates. So that's very exciting because it means that you can restore this loss. So basically, you know that you lose, the stem cell lose the capacity of eliminating the protein trash, but now you can feed them a drug and improve this and make the stem cell healthy again and the tissue healthy again. So the take-home message of my talk is that we can use simple organisms such as the fly to study somatic stem cell function, and eventually we can take all that knowledge and move them into mammals, including mice and then eventually us. And understanding how cells keep a healthy environment have very positive implication in maintaining healthy tissues, especially if you're studying stem cells, and also will have positive implication in treating diseases arising from accumulation of protein aggregates. So I included here some pictures of the fly lab if you're wondering how a fly lab looks like. So this is me next to the microscope in the lab, and basically I spend there many hours a day, of the day. And you can see this is a microscope here, and this is another postdoc in the lab looking at flies in a microscope. And you may be wondering, oh, maybe why you need a microscope? The flies are very small. These are the fruit flies that usually fly around your bananas at home. And you can see that they're very tiny, so we need microscopes. And the other thing that I wanted to show you is that we grow them in these little vials. They require a little bit of food. And then you can see here in the background of the lab many bottles having many flies. So this is a very positive things about working with flies. You can have thousands of them in the lab and do a lot of things. So with that, I want to thank my lab. This is my PI here, Henry Jasper, the rest of the lab, and also funding, and for your attention. Thank you.
So let's start with a few of these questions. The first one says, how long do you need to read a card? Let's try another one. What diseases have been linked to the inability to get rid of the protein trash?
Okay. There are many, and some of them you may heard of. So one of them is cataracts. That's something that happened to your eyes because there's a group of proteins called crystallines that they misfold when you get older, and they actually blur your lens in your eyes, so then the light cannot go through your eyes. So, I mean, that's one of them, cataracts. Other ones, you probably heard about Parkinson's disease, Alzheimer's. But these are actually not caused, they're caused by misfolded proteins and protein aggregates, but not necessarily happening in the stem cell. But this is actually happening in the neurons. But this accumulation of aggregates or protein trash can actually kill the neuron. And that's why you have people with these horrible diseases that eventually lose motor control and all of that because their neurons just die because they cannot clear the trash. So that's some of them.
So a follow-up, it's like, how do we avoid putting these protein trash in our bodies?
How do you avoid? That's a hard question. We're trying to understand that. One of the things that, like I mentioned, it happened with aging. So, and we're still trying to understand why this is happening. And one of the things that we know is that the machineries that I mentioned, proteasome autophagy, they don't work properly anymore. And so you can imagine that you can maybe treat with a drug that will increase the activity of these trash cans in the cell. But right now, as today, I will just say eat healthy, exercise, because it's good for aging itself. But specifically for protein aggregates, we'll have to wait a little bit longer to find out.
Eat healthy and exercise.
Yeah.
When I was in TV news, I just basically used to say to my reporter, we should just, every time they get up there and say, so what's new today? They said, eat healthy, exercise regularly, see you tomorrow. Because, that's most of what, if you do that, you'll kind of take care of something. Here's a question for you, Chris. Do Hep C treatments age cells?
The short answer is I'm not sure. The longer answer is there are actually Hep C treatments that work the same way as some of the HIV drugs, and those ones in the lab, we found they do in fact drive the same type of senescence and aging, but we haven't tested those ones specifically. They're just the same type of drug, so you can kind of guess that maybe, but we don't know for sure.
So we have a number of questions here that cover much of the same topic, which are, do stem cell therapies help osteoarthritis, osteoporosis, cartilage regeneration? And then there's a question about several of my relatives have used stem cell therapy to repair cartilage damage in the knee, hip, and shoulder joints with mixed results. Some excellent, some not so good. So that talks about a number of things that Karen and I deal with all the time, which is the clinics out there that are offering therapies, what we used to call stem cell tourism, because people would go abroad to get these treatments. But increasingly they're happening here in this country, and there's a recent study that showed there's something like 520 clinics in the US that are offering various therapies for cartilage repair, arthritis, osteoarthritis, and essentially what they're doing is they're taking either blood or fat, more commonly, from your own body, putting them in the centrifuge to kind of concentrate the stem cells and then re-injecting them. Now, some people I know have done this and had good results, very impressive results, others not so much. The problem is that these therapies haven't been proven. We don't know that they work. There's a reason to think why they might work, but they've never been put through a clinical trial, the kind that we were talking about earlier, where you put it through a really rigorous scientific process to make sure that, number one, it's safe, and secondly, that you know what you're doing and that you're doing it in a consistent way that's going to help the patient. A lot of these clinics offer therapies that cost $5,000, $10,000. It may not do any harm, but anytime you undergo any kind of medical procedure, there's always the risk for infection or complications. But it may not do any good either. And I think the danger with these clinics, from our perspective, is certainly that you then put at risk all the really good research that's going on. In terms of some of the specifics about osteoarthritis and cartilage repair, cartilage is really difficult to regenerate, to create, because you want something that's really strong to be able to resist all the kind of the pounding of the bones and the kind of the day-to-day wear, but it also has to be soft enough to cushion. So creating that is really difficult. But we're working with a researcher at UC Davis, Dr. Nancy Lane, and she's just got approval for a clinical trial working on osteoarthritis. And so we're really excited that that's going to be moving in a positive direction. Karen, did you want?
Yeah, there's also a lot of interesting preclinical research going on for generating new bone and cartilage tissue. So scientists are using 3D printing technology to develop scaffolds that they can seed stem cells from your fat onto, and then they can coax those stem cells to develop into cartilage tissue that you could then transplant back into a patient's knee or joint that needs new cartilage. So there is a lot of exciting research that's not ready for the clinic yet that people are pursuing that hopefully one day would help these patients.
So one of the issues with osteoarthritic joints in particular is that often there are a lot of senescent cells in osteoarthritis. And in a case like that, where you're already got those cells and they're already making those factors that might harm the stem cells or prevent them from operating normally, just getting new stem cells won't be enough unless we have some way of also treating the niche there that they're going into and making sure that that's healthy. And one of the ways we're trying to do that in our lab is by getting rid of those senescent cells.
Just to complement what Chris just said, one of the problems that happens around those senescent cells is the accumulation of these immune cells that are actually detrimental. So as you can see, like, no cell is an island in our body, and the problem is that when you mix all together a combination of things that doesn't work, the therapy is not going to work. But if you mix in the right combination of everything, including the stem cell, but everything that is around it, then it's when you have the best chance of getting a success.
I've heard that there is a high incidence of tumors with stem cell therapies. Are we making progress on fixing this?
The answer, I think, is yes. Can you hear me? Okay, this might have to do with using embryonic or induced pluripotent stem cell derived cell therapies, where the worry is that either you haven't differentiated the cells past the pluripotency point, so there might be a few cells left in there that you would transplant it into a patient, and then they could become something called a teratoma, where they make cells of all different types of tissue, where they could cause a cancer tumor and give the patient cancer. So scientists are being very, very careful about developing clinical grade stem cell lines that could be transplanted into patients that are safe. And so they're testing this first in animal models and then primate models, and then they're testing them also in humans to make sure that they don't cause tumors in humans. And so the idea is that you would transplant stem cells into a patient and not just say, oh, I hope you get better, but you would monitor that patient for 10 years or even more just to make sure that they don't develop tumors during that time.
I'll just add one thing, which is that it also depends a lot on the place where you would transplant the cells, whether a tumor is actually likely to occur or not. For example, in the eye, because it's not an environment that is pro-tumorigenic, it's very rare that we do see tumors forming if you would transplant. But then again, cells like what Chris talks about, like the senescent cells, they actually can create pro-tumorigenic environments. So other than having the best cells possible, as Karen is saying, it's also very important to think again if the place where you are putting those cells is going to tell those cells to grow in an uncontrolled manner, or it's actually going to tell those cells to do what an healthy tissue does with those cells normally.
What diseases have been linked to the inability of our bodies to get rid of the protein trash?
Yeah, I already answered.
This is one then that applies to me. Can stem cells cure mental illness? Or forgetfulness, such as depression, OCD? I think it's an interesting question. We're certainly seeing a lot of stem cell therapies that are focused on neurological conditions, on Alzheimer's and Parkinson's, Huntington's and ALS. So there's reason to believe it's possible that they could help depression. I don't know of any studies that are going on right now that are using them. But when you think about the ability of stem cells to affect so many different parts of our brain, it may well be that they can help that in some way. But I haven't seen anything particular. Karen, have you?
I think the brain is very difficult because if you transplant cells in there, there's a whole neuronal network that's going on. So it's hard to make sure that the stem cells you inject become the cell type you want and integrate properly and don't cause these aberrant connections that might, cause something like epilepsy. So I think that we need to do a lot more research before we determine whether stem cells would be able to help these patients. And also, as we've talked about on the panel, a lot of people who have neurodegenerative diseases have sick cells in their brain. So if you put in healthy cells, it doesn't mean that they're going to survive. So I think that for diseases like Huntington's disease, where it's a specific genetic mutation, that if you have it, you're going to get the disease. If we can correct that disease, the genetic mutation in these stem cells, and then have a healthy population of cells, it's a powerful model to study the sick cells and the healthy cells side by side to see if maybe we could identify new drugs that would help these patients. So for now, I think that's the powerful tool that we can use stem cells for.
Here's a question for Chris. Can you lead senescent cells towards apoptosis?
Okay, we're checking this on this time. Okay, good. Yeah, actually, there's a whole series of new drugs that are being developed that specifically generate. So for people that don't know, apoptosis is cell death. So senescent cells will stick around forever because they're basically, they're resistant to cell death. They just will sit in a tissue unless they're cleared by your immune system, and eventually they'll cause some form of disease or sickness. Recently, a group in Arkansas showed that those cells are actually resistant to apoptosis, and if they get rid of that resistance, the senescent cell will actually go into an apoptotic state, so they will actually actively die. And they're actually, that's probably right now the most successful way of killing a senescent cell that's currently available. So actually, it's a great idea, and it's actually being used right now. And it may eventually, we cross fingers, become a therapy in the next 5 to 10 years.
Is there a way to genetically modify a cell in a way in which you can replicate human traits? such as using embryonic stem cells to replicate skin with more melanin than the subject already has, or regenerate follicles that grow different hair color. I need that.
I can attempt, and then you guys can support. I definitely think this brings up the issue of designer babies. Like, now that we have this genetic engineering technology, you might have heard about it called CRISPR-Cas9. There's also other types of genome editing tools. They're like molecular scissors where you can cut and paste out sequences of DNA. So you could take a trait that maybe somebody doesn't have and give it back to them if you knew what gene was responsible for that trait. And the ethical question is, should we be doing this? I think that for parents who have diseases that could affect their children that are detrimental, like Huntington's or other types of diseases, it'd be really wonderful if they could have a child that was guaranteed not to have that disease. So you could take the embryo and correct it and remove that disease-causing mutation. But for things like hair color, I mean, I don't know, maybe you could just dye your hair. It's not worth it to do.
And that also raises a lot of ethical issues that we've been dealing with at CERN lately in terms of what kind of research do we fund? Do we fund any research that uses these new gene editing techniques to affect embryos? That can those changes can then be passed on to future generations. It's one thing to go into a child who has SCID, for example, and correct the problem there so that that child is then free of SCID. It's another thing to go into an embryo and change something so that not only will that child not have that problem, but future generations won't either. Because right now we simply don't know what kind of other consequences we may have. You may correct a gene that causes SCID, for example, but in the embryo, those developing genes may also have other things that they do, and we don't understand that. So we have to be very careful about what we do, which is why we held a workshop earlier this year at CERN, bringing together some bioethicists from all around the country to talk about this issue. And the idea was that, okay, this technology is out there. People are going to use it. Do we fund that kind of research? And our position is basically, we will fund this research to understand these techniques, to get a better understanding of how to use them in the most productive and ethical way. But we don't fund any research where the embryo is then implanted or developed after 14 days. The knowledge we gain stops at that point. And the federal government has held a similar discussion and come to the same sorts of conclusion. So these kinds of technology is really driving an awful lot of what we have to do. The technology is advancing so fast, we're struggling sometimes to keep up with it in terms of the ethics of science. So when you have the scientists here doing this kind of work, they're not working in a vacuum. They're not thinking, wouldn't it be fun to mess around with this? They're thinking, what are the consequences, the real life consequences of some of the research that we do? So these are questions that we think about all the time. Here's an easy one. Is the government funding any stem cell research? Yes, but not enough. Do you have to screen embryonic stem cells to eliminate genetic anomalies?
Yes. It's a very simple answer, yes. And actually from when you are generating stem cells, the amount of cells that you generate and the actual, the proportion of those that are actually fit to be transplanted is very small. So it's a very rigorous process. And scientists who do it, and especially if you do it in people, you know that that's a step, there's many lines which you can't cross. So we do have to generate enormous amount of cells to find the ones that we are absolutely sure are not containing those mutations that can cause the problems.
Well said.
A number of questions are, what is the status of research on Alzheimer's disease and how close are we to meaningful Alzheimer's treatments? Karen, do you want to start with that one?
That's a hard question. Alzheimer's is not just one disease. It's people with Alzheimer's have many different symptoms. It affects people at different ages, and I think that right now it's difficult because there are not a lot of, at least stem cell treatments that are being tested in clinical trials for Alzheimer's. People are still focusing on developing drugs, but there hasn't been one that has been shown to be really effective yet. So a lot of pharmaceutical companies are putting in a lot of effort to try to tackle this problem. And I think on the research side, scientists are still trying to produce effective models, whether they be mouse models or stem cell models of Alzheimer's. So we're not quite there, and I can't put a date on it, but.
And I think another problem is that we're not even really sure what causes it. I mean, there's still a lot of debate about the buildup of plaque in the brain. Is certainly one indication of Alzheimer's, but we're not sure if that's the cause or a consequence. And part of the problem is by the time people have developed the symptoms, this has been going on for many, many years. And so it may be that the people we're trying to treat may be past the point where we can effectively really do anything, at least with existing medications and therapies and approaches. It may be that we need to develop approaches and identify people who are at risk and maybe get to them earlier. But again, that's complicated. Alzheimer's is one of the questions we get asked the most, and it's also one of the trickiest ones because in many ways we're still just kind of stumbling through understanding what it is.
I just wanted to add that we have an expert in Alzheimer's research in the back, Claire, so if you have more questions for her, you can ask her. So she knows a lot of Alzheimer's, so you can ask a specific question to her.
And there was another question, the same sort of question about Parkinson's disease. And again, Parkinson's disease is very complicated. It's basically caused by a lack of dopamine-producing cells in the brain. But just treating that doesn't seem to kind of change the course of it. And there's a lot of research going on all over the world. I mean, at CERM, I think we have something like 23 different projects that we're funding in Parkinson's research for more than $56 million. Some of it's going on here. There's Xiaomin Zheng here at the Buck is doing some really fascinating work. And we're hoping that at some point we'll be able to kind of progress into clinical trials. But really it's very slow because, again, it's a very complex disease. And anytime you're going into the brain to try and create changes, you have to be careful about all the other kind of consequences that they may have. And also, you really need to think about what are you trying to do? Because we have a Parkinson's member on our board, and he was just talking about when people ask Parkinson's patients, okay, we're going to help you, we're going to get rid of all this dyskinesia, these muscle movements, and he's saying that wouldn't even come in the top 5 of complications that most Parkinson's patients talk about. For them, it's fatigue, it's insomnia, it's depression. There's all these other things that Parkinson's brings on, induces, causes, that no one seems to be addressing. And so when we talk about treating a disease, we often try to isolate one element of that and say, we'll fix that. And the real picture is much more complicated.
May I ask a question?
Absolutely.
Both my parents had late onset Parkinson's. I'm shaking. What's my disposition to that possibility?
I'm not a physician, I wouldn't dare.
Please repeat that.
Yes, the lady in the front saying both her parents had late onset Parkinson's disease, and so she's wondering what are the odds, what are the risk factors for her?
So my dad actually has Parkinson's disease, and one thing that you can do is to get your genome sequenced, and so he did sequence his genome through 23andMe, and he did not have any of the genetic predispositions to Parkinson's. So there's specific genes that have mutations that give you a higher chance of having Parkinson's, and he did not have any of those. So that gave me a sense of peace of mind. So that's one option. And Parkinson's is unique where there are some genetic risk factors, but it's also a sporadic disease, meaning that environmental factors could cause genetic mutation to build up over time. So it's not necessarily that you will get it, but there are ways to give yourself more knowledge.
So there are ways to possibly prevent it?
Right now, we don't know of any way to prevent Parkinson's, but there are both drugs and surgeries that you can have that delay the symptoms, so that way you live longer and have a healthier life.
There are a number of questions about clinics, such as, is it advisable to use stem cells for arthritis? A company like Nirvana out of Sacramento does this. And the FDA has not approved stem cell treatments. However, there are treatments now that you can go to. But how do you know who to trust, such as going to other countries for treatments? We touched upon this earlier, this notion of stem cell tourism and the fact that there are many clinics out there that offer things that really haven't been approved. And particularly you can go abroad for therapies. I mean, many clinics here will have a branch in Mexico, for example, where they'll do the work here, the initial analysis here, and then you go to Mexico for the treatment. And the problem is we've seen some real complications from people who go abroad for these treatments. There was a woman who went to the Dominican Republic for some just purely cosmetic procedures, taking stem cells from her fat and using them to kind of plump up her face, make it look younger. And when she came back, she couldn't open her left eye, and every time she did, it clicked. And so finally she went to see a dermatologist, and he found that she had bone growing in her eyelid. Because they'd taken some of these fat cells and injected them into her eye. And these fat cells have the ability to be turned into cartilage or bone. And they'd also used kind of a stimulant, a growth factor, to kind of promote that they wanted to get these stem cells to be even more fluffier, if you like. But it stimulated this bone growth. And so these are the kinds of complications that can happen. We've heard of cases where patients going to Russia have died as a consequence of some of the therapies they received. These are rare, but they can happen. And so this is the kind of the warning about going to these clinics, going to these places that don't have an approved or licensed therapy. We're also seeing for me, a particularly disturbing thing, which is these clinics that are offering therapies for arthritis or knee joints or hip problems, are saying that the same treatment, the same kinds of approach can work for things like autism, like Alzheimer's and Parkinson's. And there's just no way of thinking that what they're doing could benefit those diseases. And the worst thing is that it's preying on the sense of hope that the patients have. They're running out of other options, they're running out of time. So they will turn to anything. And these clinics are giving them a real false sense of hope that what they're going to get could help them. And they're also taking their money. And so this is something that I think that we really need to be very mindful of. How successful are stem cell implants to grow a new lens instead of having an artificial lens implanted after cataract surgery? Yes. How successful are stem cell implants in growing a new lens instead of having an artificial lens implanted after cataract surgery?
So we have grown lens in the lab, in the plate. So it's possible to grow. We have not transplanted, and I do not know from myself of cases in which it has been transplanted. But as you've heard, stem cells can make any cell. They can be whatever they want. And certainly they can do lens. And we have made lens here in the lab when we are making every other cells of the eye. So it is possible.
So selfishly, I'm going to read this one last. Karen, who should citizens talk to in the government to make changes to stem cell therapy approval? And does CERM handwrite patients a list or give patients a list of legislators?
I will hand that question off to Kev.
Thank you.
Yes, you can talk to us.
We're a state agency, and so we're a political entity, and so really we can't, as a matter of law, as a matter of course, give people letters and say, send this to your legislator, send this to the FDA, here's a list of people you can send it to. So what we do with the Stem Cell Champions campaign is we give you the materials you need so you can write that kind of letter yourself, and that you can direct it to whoever you think is most appropriate. And often it's something as simple as just your own congressperson, our own US senators, even local legislators in the state assembly, or just to the FDA. You don't really need an address, you can just address it to the FDA commissioner, Dr. Robert Califf, and say, what do you think? He's really good, actually. I think he's quite new, but he's come in and he's really trying to change things. But it's a huge organization. I mean, $1 in $4 in the US economy, $1 in $4 is touched by the FDA on some level or another. You talk about food, you talk about medicines, cosmetics, every single thing. It's enormous. And so for one person to be able to kind of make the changes that we need there is going to be very difficult. He's going to need all the support that he can get from people like you, from all of us, to make these changes. And so if you write to him, if you write to your local legislators, that would be something that really starts out. I would like to point out that we have some clipboards at the back. So as you're leaving, you can sign your name to how to become a stem cell champion, and we'll get to you.